All right, ladies and gentlemen, welcome back. So today we're going to pick up talking about enzymes themselves. So we've kind of been mentioning the importance of enzymes when we look at chemical processes, metabolic pathways in organisms. So let's just make sure we're all on the same page and know what these enzymes are. So when we talk about enzymes, they're biological catalysts, right? So they are chemical agents that will speed up chemical reactions. But the cool thing about them is that they don't get consumed by the reaction itself. And so frequently when you see enzymatic reactions written, you'll actually see the enzyme written over the yields arrow because it isn't a reactant, but it's not a product either. So it's involved in the reaction, but I don't consume it, which is a good thing because that means I can reuse it. And that is going to be important because we have to build these enzymes. They are catalytic proteins. That means your body has to create them. So we have to take the amino acids that you eat, rearrange them, put them together to make these enzymes. If we had to consume an enzyme every time that they were necessary for a chemical reaction to occur, you'd never be able to stop eating those proteins in order to gain enough amino acids to constantly build those enzymes every single time a chemical reaction had to happen. And that's assuming you could consume enough of those amino acids, period. So when we look at enzymes, they can kind of be just about anywhere that a chemical reaction is necessary. We will see that enzymes tend to be grouped into complexes. Sometimes they're incorporated into your cell membrane or into the membranes of other organelles. Other times they're just kind of hanging out waiting to be encountered in your cytoplasm, although the efficiency on that end is considerably lower. And so most cells have evolved ways to get those enzymes in a particular location so that they can more efficiently catalyze the chemical reactions that the cell needs them to. So let's go back to this idea of thermodynamics, right? So why do we need enzymes at all? Why do we need these biological catalysts, right? Well, if I'm looking at a reaction that is exothermic or exergonic, as we've been using that term, um, that means that regardless of the chemical reaction, I need energy to be taken in to take those reactants, break their bonds, and rearrange them, right? So every chemical reaction needs energy in. When we break the bonds in the reactants, every chemical reaction releases energy when we go to build the bonds in the products. The question becomes, how much energy do I need to put in as opposed to how much energy do I get back out? And so what we're really talking about is the difference between the activation energy needed, right? That EA, that free energy of activation, as opposed to what is the delta G? What is the change in free energy from reactant to product? If I get a release of excess energy when I make those products, then that reaction is probably an exergonic and it's going to happen spontaneously. And so we probably aren't going to necessitate enzymes in that situation. However, if I'm looking at the opposite, where the amount of energy required to break the bonds in those reactants is actually less than the energy released when those products get formed, that's when we're talking about endergonic reactions, and that's where the necessity for enzymes comes in. So enzymes speed up chemical reactions, right? That's the definition of a catalyst. How are they doing it? Well, all they're doing is lowering the amount of activation energy necessary. They don't get rid of the need for activation energy entirely. They're just going to lower the required input. So think of it kind of like you're buying your first car, right? And so you're working that part-time job at McDonald's, Wendy's, Dunkin', Walmart, whatever the case might be, right? And you're working minimum wage. So you've got a finite amount of hours you can work as a teenager and a finite amount of money you can generally make from those part-time jobs. So if it were up to you to have to gather all of the money to put to buy a car outright, it would probably take you forever. But in steps grandma, right? And grandma says, so long as you're maintaining good grades and you're going to your job regularly like you're supposed to, she's going to help you out. Grandma's kind of like the enzyme. So she's not going to give you all of the money to buy that car, but she's going to give you some amount of money to help you get there faster, right? So maybe that car costs you, I don't know, $2,000. She's going to give you 1,000, right? And so she is helping by lowering the amount of energy, or the amount of money in this case, needed to get you to that end goal faster. That's what the enzymes do. So you still require the input of ATP, you just require less of it. So a couple of terms, just to make sure we're on the same page here, right, as we sort of talk through how the enzymes are going to actually work in lowering that activation energy, we need to make sure we know a couple of components here. So this is a basic enzyme cycle, right? So you've got your substrates here. Those are the molecules that that enzyme is designed to work on. Keep in mind that enzymes are substrate specific. They either work on just a single substrate or a very small, very similar class of substrates. Those substrates are very specific to the enzyme based on the way that that enzyme is built, right? Remember, these are proteins, so those proteins go through those series of protein folds in order to give it its three-dimensional shape. Part of that is the shape of its active site, okay? And so that active site shape is what's going to drive what kind of substrates it can fit, all right? If that's a very tight, very specific shape, then maybe it only works on a single substrate. Other times it will work on some things that are very similar just based on the way that the protein is put together. When we put the substrate plus the enzyme together, that enzyme literally gives those substrates a bit of a hug, a bit of a squeeze. We'll talk about this idea of induced fit. But the combination of the enzyme plus the substrate, we form an enzyme substrate complex, and that's when that enzyme is able to actually help to lower the activation energy and catalyze the chemical reaction. So here's that idea of induced fit, right? So if those enzymes are going to lower the amount of activation energy, when that substrate comes in, we need just enough wiggle room to get it in there and be able to lock it down tight. And so that's this idea of induced fit. So by bringing those substrates into that active site, the enzyme will actually squeeze on and clamp onto that substrate to finalize that connection, to finalize that substrate complex. And that's the idea of an induced fit. Once that induced fit is there, now the enzyme can actually do its job. And so once that induced fit is activated, substrate is now bound to that enzyme. That enzyme has the capacity to speed up those chemical reactions really in one of four ways. Um, One is to simply ensure that the orientation of those substrates is correct. What that means is if I'm doing something where I'm taking two monomers, for example, and I need to build them or put them together, by orienting those substrates correctly into that induced fit idea in that active site, I can get those substrates really nice and close together and be able to facilitate the bonding of those monomers by just having them really close together, right? So it's not like I'm not going to need energy in order to finalize that bonding, but by having them oriented in just the right way, I can lower the amount of energy I need to put in order to get them to link together. Another way that we can speed up chemical reactions is by actually straining the bonds in the substrate. So if it's not an anabolic process, but a catabolic one, by putting those substrates into that active site and inducing that induced fit, I actually put them together and put excess strain on the bonds that are currently holding that molecule together, so it makes it easier to break, right? If I were to hand you a pencil, right, and tell you to snap it in half, it's going to take some serious energy probably to do that. But if I put some cracks in that, because I've already sort of helped, it's going to crack a lot easier. And so the enzyme, by squeezing and putting some extra pressure and extra strain on that bond, allows it to break easier, break with less energy. We can also work on just providing a favorable microenvironment, so getting other competing molecules out of the way that might inhibit the ability for, say, something to bond together by giving it just a favorable pH or favorable salt concentration or whatever the case might be, shielding it from other molecules it might also want to interact with. I can help to facilitate the chemical reaction that way. And that all sort of ties into this idea of just being able to covalently bond them to the substrate, right? Adding things to that to, again, facilitate the chemical reaction. So remember that I mentioned this is a general diagram of an enzyme cycle, right? It's a catalytic cycle. Because that enzyme doesn't get used up in the reaction, once the products get formed here, whatever those products may be, those products get, in the process of them forming, adjust the bond in the active site and allow them to be released. Then our enzyme is back open with that active site available again to be reused once again. And like I mentioned, the efficiency in the fact that we can continue to reuse the same enzyme over and over again is a really big deal to ensure that you aren't required to be taking in all sorts of material in order to just build the enzymes. Eventually, the enzymes wear out, and so you will have to replace them, but it's not going to be a once and done thing. So here's the thing with these enzymes, though. They're proteins, which means they've gone through protein folding, but the bonds that are holding that three-dimensional shape of that enzyme together are very much subjected to the environment that they're in. They're very sensitive to that. So we can have environmental shifts that occur within a living thing that can then affect that enzyme's activity, right? And this goes back to the whole idea of why do organisms expend so much energy to maintain homeostasis? This is why. They need to ensure that the environment internal to their cells is stable enough to ensure that those enzymes are stable and capable of doing their job at as best a rate as possible, because all of those chemical reactions are driving how that organism functions. So when we look at enzyme activity graphs, they tend to look like these, where you'll see sort of an increase in the rate of the reaction. It'll peak out where it's optimal, where it's doing its job the absolute best, converting the product substrate to the product as efficiently as possible, and then you'll also see a dip on the other end. So most environmental factors that are going to affect the enzyme activity are things like temperature, pH, salt concentration. Those are usually the three big ones that we talk about. When you look at the ones here, we're looking at optimal temperature versus pH, right? These are just two examples of these graphs. And again, because they're proteins, if I get that protein, if I get that enzyme outside of its best conditions, it starts to lose its three-dimensional shape. So when I'm looking at the up curve here or the down drop on the other side, the enzyme's still working. It's just not working as efficiently as possible because the folding isn't as complete or as tight as it could be. When we're at that optimal temperature or optimal pH, then the enzyme is folded to the absolute best it can, and so it is working as best as possible. When we start getting on either side of that, the proteins start denaturing. They start losing that functional shape. Once we get so far outside of the optimal conditions, like we see here in the pH, where there's absolutely no data at all, there's no reaction happening here around pH 4 or 5, that's because those proteins have completely denatured. They are back to just straight chains of amino acids, and they're not doing anything. All right. So while I'm looking at the increase in pH here, the folding's getting better and better and better and better and better until it's absolutely perfect, at least as perfect as nature has made it, where it's facilitating that reaction as best as it can. And then I start dropping back down again as that pH continues to climb, and I'm losing that functional shape again. I'm denaturing again. You can also have other ways to regulate these proteins, or just come in contact with other compounds that will affect the way that that enzyme works. And these are the ideas of enzyme inhibition. Some things, like cofactors and coenzymes, are non-protein sort of enzyme helper molecules. They tend to be inorganic things, metals, or if they are organic, then we talk about things like vitamins. So when you talk vitamins and minerals and why do you need them in your diet, why do you take that multivitamin perhaps in order to get them, is because a lot of those vitamins and minerals will act as these cofactors or coenzymes. So they attach in places like perhaps in the active site to enhance that induced fit of its substrate, to allow the binding of that substrate to be even better than it would if that cofactor or coenzyme didn't exist. In a lot of cases, the absence of that cofactor or coenzyme doesn't mean that the enzyme doesn't work at all. It's just not as effective as it could be. And in other cases, the lack of them means it's not going to really bind at all. Give you an example of one of these cofactors is the binding of oxygen to hemoglobin, right? Hemoglobin is the major protein in your red blood cells, okay? And the whole point to that hemoglobin is to bind the oxygen to your cells. Well, oxygen can bind to hemoglobin. It does an okay job of it, but if you have enough iron in your body, iron acts as that cofactor. And so having the iron plus the oxygen plus the hemoglobin means everything binds much better, much tighter, much more effectively. And so you're able to carry much more oxygen around your body and do it much more efficiently if the iron is present. We've got two major classes of inhibitors here. And so we've got competitive inhibitors as opposed to non-competitive inhibitors. And a competitive inhibitor will directly compete for the binding of that substrate to that active site. So it will bind to that active site, preventing the substrate from actually binding. It literally competes, literally gets in the way. A lot of times when we see competitive inhibitors, those sort of bounce in and out of that active site because they're not perfectly shaped. And so you won't necessarily completely turn off the ability for that enzyme to do anything, but you'll certainly slow down the rate of reaction. So you'll often see graphs in the sense of what it looks like without the competitive inhibitor, and then when you compare it with a graph of the action with a competitive inhibitor present, that graph will be much lower, right? You're producing less product with that inhibitor present. Your non-competitive inhibitors will bind to a secondary site, what we call an allosteric site. We'll talk a little bit more about allosteric regulation in the next video. But what that does is the binding of that non-competitive inhibitor, when it binds to that allosteric site, manipulates the folding of that protein and ultimately changes the shape of the active site. And so when we see this, because it's changing the shape of the active site, it's not the right shape anymore for the substrate can't bind. You'll often see a much tighter association of non-competitive inhibitors than you do competitive inhibitors. And so as a result, a non-competitive inhibitor has the tendency to shut that enzyme down completely and just completely stop the ability for that chemical reaction to occur until the binding of that inhibitor gets released. So why do we care about inhibitors, period? Well, we do use a lot of them in just experimental design. We want to see what an enzyme does, and so we shut it down, right, and see what happens. So they definitely provide some insight into the function of the enzyme and what it's doing in that cell. We also see that nature has sort of developed toxins and poisons that are inhibitors. So you come in contact with something like cyanide. Cyanide's a poison. It is an inhibitor, I should say. So it binds to those enzymes, prevents you from being able to do your job, ultimately kills you, all right, because you're not able to finish the metabolic pathway that that enzyme is a part of. They will regulate enzyme activity, so selective inhibition. We'll talk a little bit more about that again in the next video. Just think about it. If I have all of these enzymes functioning and around all the time, how do I regulate when I use them and when I don't, right? Because sometimes I want to keep a certain level of concentration of a carbohydrate in my body. I don't want to break it down. Or I've spent so much energy building this molecule, but now I have enough of it. So why do I want to keep building it? That's using resources and energy that's not necessary, right? So we want that selective inhibition to be able to regulate those enzyme activities. And a lot of that is done by non-competitive inhibition, and we'll talk again more about that in the next video. We also take a huge advantage of these pathways and the enzymes that regulate them by being able to create drugs and medications that are similar in shape to that substrate or just act as the inhibitor itself. If you ever listen to drug commercials, the one that always pops into my head is MAOI inhibitors, right? It's an enzyme inhibitor. When we look at things like depression medications or anxiety medications, a lot of them will work as inhibitors to prevent those neurotransmitters from being digested and broken down in your brain so that they stay around longer and can continue to perform their job of making you feel better. So a lot of that has to do with understanding the enzymes that are present, what they work on, and then how can we prevent them from doing their job.